In the last video, we talked a lot about the structure of the cell membrane and the two forms of cell transport that allow certain things to move in and out of the cell. So for a quick review, you can see the cell membrane here, and you can see that there are two layers, and they are layers made up of phospholipids. So we would refer to the structure as the phospholipid bilayer. And you can see that there's also some proteins embedded or stuck inside of the membrane that help allow certain things in and out. Passive transport is a way in which things can move in and out of the cell with no energy. Active transport requires active energy, right? So active needs ATP. ATP is what powers active transport. Passive transport moves things from high to low, and active goes from low to high. But let's look a little bit more at the passive transport, the ways in which things can move into and out of the cell with no energy. So typically speaking, molecules like to spread out. They don't want to stay next to each other. We've seen this when we make sweet tea, right? If you put a tea bag, right, into a pitcher or into a cup or into a beaker with water, you'll watch the color, the brown color, the tannins from the tea slowly spread out until the whole solution or until your whole container is a brown color, right? Or if you've ever made Kool-Aid, right? And so you dump in the Kool-Aid powder. The Kool-Aid powder is initially highly concentrated, right? So it's all really close together, and you dump it into the water. Well, eventually the Kool-Aid will spread out, and then your whole cup or pitcher is filled with that color. And so molecules naturally want to go from high concentration to low concentration. They want to spread out to reach equilibrium, which equilibrium means that these molecules want to get as far away from each other as possible so they're all equal distance apart. And no energy is required to do this. If you had a glass container filled with cologne or perfume, the cologne, the perfume, those molecules that smell good are all highly concentrated in that container. If you were to drop that glass container and were to smash on the floor, the highly concentrated perfume and cologne would slowly start to spread out throughout the room. And so if you were on the other side of the room, you'd eventually smell the cologne because those molecules are going to slowly spread out until they reach equilibrium. Now, we need to introduce some vocabulary. Now, let's say we've got some salt. All right, and we want to dissolve it in some water. We all know that salt dissolves in water. We all know that sugar dissolves in water, but now let's use some scientific vocabulary. Now, the salt and the sugar that we're going to add to the water, we refer to it as the solute, okay? And the solute is what gets dissolved, right? And so we add the salt to the water, and initially it's going to be really highly concentrated, right? You can actually see the salt crystals, right, as they drip or drop to the bottom of the container, right? And so you may end up with a bunch of salt crystals at the bottom, and they're all highly concentrated. Now, they will eventually dissolve in the water, so maybe you pick it up and you swirl it around, and then after a while, all of the salt crystals dissolve, right? Well, really what happened there is the solute, or the salt or sugar that you added to the water, it dissolved. And so we would say that the salt and the sugar, they are your solute. They get dissolved. And in our example here, we were dissolving them in water. Water in biology is considered the universal solvent. Water does a really good job of dissolving things. And what we end up with is salt water. And so this would be our solution, right? And we could kind of think about the salt water as if water and salt were perfectly mixed together, right? The salt molecules are as far away from each other as they could possibly get. So we had gone from high concentration to low concentration. We dissolved our solute into our solvent. Now, with passive transport, there are three main types. We've got diffusion, and this is sometimes referred to as simple or complete diffusion. We've got facilitated diffusion and osmosis. Now, what is diffusion? Well, diffusion, right, I'll give this a number, right? So we'll call diffusion number 1, is what you see right down here, okay? We've got a lot of molecules, right? And they are highly concentrated, right? So they're a high concentration right here. You've got lots of these little orange dots, and they are going to passively diffuse into the cell from high concentration to low concentration and that's simple diffusion. No energy necessary. These molecules want to do that. The next one would be facilitated diffusion. I'll use a different color, right? So facilitated diffusion, it's when molecules need help. And so when you see this word facilitated, you could also think of this word as help, right? So it needs help. And there's two reasons why. One of which might be that the molecule is charged, right? And so maybe it has a chemical characteristic or chemical property that does not allow it to easily diffuse to the membrane and needs some help, right? And it gets help from a carrier protein. And so we're still going from high to low, but we need some help. And the help is provided by a protein, right? A protein channel. Sometimes the molecule is just too big, right? So in the case of glucose, glucose is a huge molecule, right? C6H12O6. That's 24 atoms, right, in that one molecule of glucose. And it's huge, right? So that glucose molecule is too big to easily diffuse through the membrane. So it needs some help, and it gets some help from a carrier protein. The glucose molecule binds to the carrier protein, and the carrier protein changes its shape. Now, again, we're still moving the glucose molecules from high concentration to low concentration, and so no energy is needed. And then the last one is osmosis. And I really should have done osmosis in blue because osmosis is the movement of water. And so down here, right, we've got water molecules, and water molecules are really small, right? H2O, we're only talking about 3 atoms here. And water can easily find its way through the plasma cell membrane. And water would still want to go from high concentration to low concentration. Now, in some organisms, the cells need to get a lot of water in or a lot of water out quickly, and they'll actually have some protein channels there to help. And so that would be an aquaporin. And since a protein is helping, this would be an example of the facilitated diffusion of water, right? Or facilitated osmosis. Now let's take a look at some situations and try to figure out where the water's going to move, right? And so in biology, a lot of times we're really concerned with water. Where's the water going to go? And so here we have 3 cells. Now, how do I know what kind of cells these are? Well, these are plant cells. How do I know? Well, look at their shape, right? They kind of have that boxy, rectangular, square shape, and that's because plant cells have that outside layer, the cell wall, and a cell membrane, right? So the cell wall would be this square shape, and then the thinner line on the inside, right, would be the cell membrane. And so here we've got a plant cell that's shrinking, right? Water is moving out of the cell. Now, why would this happen? Well, the movement of water, again, is going to be controlled by concentration. And so if water is moving out, well, then we can probably take a guess. And we can say, you know what? The water concentration inside of the cell was probably pretty high. And the environment or the solution that the cell was floating around in probably didn't have a lot of water. So the water moved from high concentration to low concentration. In this case, our plant cell would shrink. Now let's look at this second example, okay? And we can see here that some water is going out and some water is going in, and maybe they're moving in and out at the same speed. So we're going to have no change, right? And so what's probably going on here is that the inside and the outside, the amount of water is probably equal. So we could say that the concentration of water inside of the cell and outside of the cell is equal. And so we're going to have no real change, right? It's not going to get bigger. It's not going to get smaller. It's going to stay the same. And in this last example over here, we can see that we've got a lot of water rushing into the cell. Oh, hey, look, our vacuole, right? The organelle where water is stored inside of plants. And this vacuole, as it fills with water, is going to push against the outside of the cell, causing it to swell. And you can kind of see the cell wall here kind of pushing on the outside. Now, why would water go rushing into the plant cell? Well, remember, water wants to go from high concentration to low concentration. So maybe this plant cell was dehydrated, right? And it just rained. And so water from the soil quickly rushed into the root cell, right? Or the plant cell in the soil, right? So water's moving from high concentration to low concentration. Now, are there still sugars and salts in the environment surrounding these cells and inside of the cells? Yes, absolutely. But at the end of the day, and especially for our class, when trying to figure out what the cell is going to do, is it going to shrink? Is it going to swell? Or is it going to stay the same? We really just want to figure out, what is water going to do? And the movement of water is known as osmosis. Whereas the movement of the sugar and the salts, that's diffusion. Let's take a look at some more vocabulary. Okay, I know there's a lot, but let's look at one piece at a time. So here we have a red blood cell, right? The cells that we find in our blood. And we have taken the cell and we've put it into a beaker. And it looks like some water's going out of the cell and some water's going in, but it looks like it's equal. And this cell is not going to change shape. It's going to stay the same. And we call a solution or an environment that has the same concentrations of solute and water as the inside of the cell, we call that isotonic, right? And so nothing is going to happen to the cell. The cell is at equilibrium. Now, if we look at this next red blood cell, whoa, that does not look like a good day. If that was happening inside of you, it would not be good. So what's happening? Where's the water going? It looks like water is rushing into the cell. We got a lot of water moving into this cell, and what's happening? Well, the cell is getting bigger, right? It's swelling with water. Now, why might that happen? Well, if water is flowing into the cell, it means that the environment had a lot of water, right? So we had a high concentration of H2O, right? There was more water in the environment, and the cell started off with less water. And so the water wants to rush inside of the cell, and that's why it's swelling up. Now, we would call this environment hypotonic. And there's a reason we use these terms, hypotonic. Hypo means below, and tonic is referring to the strength or the amount of solute, right? The sugar and the salt. And so if we were to really look at this, right, if the environment is really watery, if there's a lot of water in the environment, we would say that's a dilute solution. And what does that mean? Well, diluted means that it's mostly water and there's a very small amount of solute. And if there's low water inside of a cell, that probably means there's a lot of other stuff, a lot of solute inside. And so we would say the solution, the environment, is hypotonic. There is less solute in the environment than on the inside of the cell. And I always just think hypo. So a hypotonic solution is going to cause our cell to swell up like a hippo, right? Hypo. No, I'm not an artist, but this has helped me over the years. So I think hypo, if I take a cell and I place it into a hypotonic solution, that means that that environment or that solution has way more water or a higher concentration of water than inside the cell, and the water will come rushing inside of the cell and cause it to swell up. Now, what about the final example here? It looks like all of the water is leaving, right? And so the water is rushing out of the cell. Why might that happen? Well, it probably meant that there was a lot of water in the cell to begin with, and it meant that the environment probably had a low concentration of water. So the water wants to go from high to low, so the water is going to try to escape, and that's going to cause the cell to shrink. Now, we can also say that this solution is highly concentrated. There's a lot of solute, right? So maybe each one of these yellow dots represents sugar or salt. And so the solute here, right, is highly concentrated, so that means there's not a lot of water here. And that means the water is going to get sucked out of the cell. And so hyper means above. And again, tonic is referring to the strength of the solute, which would be sugar or salt. Okay, and so hypertonic, things tend to shrink, right? And so I think like hyperactive, right? So somebody who's hyperactive, they're always on the run, right? So maybe somebody who's, always running around, they're going to be really thin, right? They're going to be really, really like slender individuals because they're constantly, bouncing around and, they're expending all kinds of energy, right? And so they're going to be really, they're going to shrink, right? And so that's why I think hyperactive. I'm a hyperactive person, right? And so I'm rather skinny, right? And so I think shrinking, okay? Hippo, hyper, shrinking. Now, hypertonic solutions are what we use to make things like beef jerky or jams and jellies, okay? If you want to make beef jerky, right, you want to preserve that meat, you're going to cover or rub the outside in a salty solution, right? Or you're going to marinate the meat in a sugary or salty solution. And what that means is the salt and the sugar is going to suck the water out. And so the meat is going to shrivel up, and then you end up with beef jerky. Now, to make jams and jellies, we take fruit and we put it into a mason jar or into a container. And we add a lot of salt. We add a lot of sugar. And that sugar draws the water out, the fruit shrivels up. And the reason that works and the reason that actually preserves the food is it also pulls water out of bacteria, right? And so if you put a bunch of salt on the outside of meat, yes, it's going to pull the water out of the meat, but it's also going to pull water out of bacteria. And if you pull the water or remove the water from bacteria, they won't be able to survive. Same with your fruit, right? The strawberries that you're making your preserves, jellies, and jams with, there's going to be bacteria and yeast on the outside. So you wash them and then you put them in a sugary solution. The sugar is going to draw the water away from the bacterium and dehydrate them so they will not be able to survive. Let's take a look at a few examples and let's see if you can figure out what's going to happen. So here we have a cell. And we need to figure out what's going to happen. Okay, so solute, anytime you see that, just think sugar or salt. Don't excuse my handwriting. And then anytime you see solvent, just think water. Okay, and so it looks like we've got a lot of sugar and salt inside of the cell and not a lot of water. But if we look at the environment, it looks like the environment doesn't have very much sugar or very much salt. And it looks like it's got lots and lots of water. Because remember, water is our universal solvent. So what is the water going to do? The water is going to want to go from high to low. And anytime water rushes into the cell, what's going to happen? Well, our cell is going to swell. It's going to increase in size as it absorbs water. Now, why would it do that? Well, because water wants to go from high to low. Now, how could we describe this environment? Well, we would say that this environment is hypotonic. And remember, hypo means below, and tonic is referring to the strength of the solute, right? And so hypotonic means the environment is less than the inside. All right, let's take a look at another example. So here I've actually given you numbers, right? I've given you measurements, concentrations. So again, the solvent is water, and the solute could be all kinds of things. It could be sugar, it could be salt, it could be ions, it could be carbon dioxide. It's just anything that's dissolved in water. Well, where does the water want to go? So we've got more water inside now, and so the water is going to want to go from high to low. And if water leaves the cell, what's going to happen? Well, our cell is going to shrink and shrivel up. So we're going to have a cell that shrinks and decreases in size. And how would we describe this environment? Well, this environment would be hypertonic, or this solution that the cell is floating around in would be hypertonic. And what does that mean? Well, it means that the outside is higher. The solute strength is higher outside than it is on the inside. 60 is above or higher than 30. So hyper means above, and 60 is definitely above 30. Okay. Now the sugar and the salt does want to go inside, but remember, the cell membrane is selectively permeable. So the cell does have some control over this, and the cell might not want all of that salt and sugar to come rushing inside. Let's look at one more example. So here I've given you some more percentages, and I've also given you a diagram. So what's going to go on here? Well, remember, we're only really concerned with water. So inside of our cell, we've got a water concentration, because remember, solvent is water, is 35. So we got 35% water on the inside. And our environment, or our solution on the outside, is 50-50. So we got 50%. So water is going to want to go from the solution or the environment, and it's going to want to make its way into the cell, which is going to mean that this cell is going to get much bigger. Now, this is also why we can't take a freshwater fish and put it in the ocean. And we can't take a saltwater fish and put it in a freshwater fish tank. Okay? If I take a saltwater fish and put it in a freshwater aquarium or fish tank, there's going to be way more water in the environment, way more fresh water in the environment that saltwater fish is used to. And the fresh water is going to rush in, and it's going to cause that poor saltwater fish's cells to swell up. Now, if I take a freshwater fish and put it in salt water, we all know that the salt is going to slowly draw or suck the water out of that freshwater fish, and that freshwater fish cell is going to shrivel up. My high school biology teacher always just told me, just think salt and sugar suck. They suck water out of cells. So if you want to make a cell small, just put it in a really, really, really sugary, salty solution and the water will leave. If you want to make a cell swell up, put it in fresh water.